continue with Eva Ilioska from Cambridge, optimizing connectome-based reservoir computing.
So our understanding of how the brain structure and function are connected largely comes from observing differences in brain structure across individuals or groups of individuals, where by following injury, for example, and then systematically linking these differences to brain function or behavioral outcomes. But how does the relationship between brain structure and cognitive function emerge in the first place? What exactly in the structure of the brain contributes to the variety of individual differences we observe, or what contributes to cognitive function turning into dysfunction? An important piece of the puzzle is looking at the brain as a network. Connectivity patterns have an important role in brain function and performance, and we know that the brain follows certain wiring rules. For example, the presence of highly clustered and segregated modules supports specialized information processing, while the efficient wiring of high-degree hub nodes supports the information integration across the specialized modules. These network features are correlated with cognitive functions and diagnostic groups, but there is no clear mechanistic link between the structural network properties of the connectome and cognition. In a very old paper, Massimini and colleagues found that whether we have conscious experience depends on how and if a signal propagates through the brain. They stimulated participants with transcranial magnetic stimulation during wakefulness and sleep. And they found that during wakefulness and REM sleep, the signal evoked a local response, and then in sequence it spread through other cortical locations, producing a long-range patterns of activation through time. While during non-REM sleep, after the initial spread, the signal remained local, indicating a potential loss of cortical integration. When we look at just the relationships between the structure of the brain as a complex network and the cognitive function that emerges from it, we ignore the way the signals propagate through the complex network, and in a way we skip a step in our inference. This indicates that the computational capacity of a network, or how well a network allows for activation to spread through its nodes and edges, might hold the key to understanding how different networks' properties impact the performance or behavior output of the brain. To address this question, we need to be able to experimentally manipulate how the connectome is structured and how, depending on its structure and dynamics, it allows for the propagation of inputs through the network with the goal of learning to perform a task. In other words, how does the network leverage its topology in order to perform a given task? Reservoir computing makes it possible to model how recurrent neural circuits extract information from a continuous stream of stimuli by mapping them in a high-dimensional space. The reservoir system has three parts: the reservoir itself, which is a recurrent neural network with fixed weights. The fixed connections of the reservoir resemble structural brain connectivity, which makes it a particularly suitable model for studying the impact of structural brain organization on network dynamics and computational capacity. And the reservoir receives its inputs through the input layer, which is a set of nodes from which the input signal starts propagating through the network. And lastly, the readout layer, which is also a subset of nodes. And the weights of these nodes are trainable, and this is the only place where the learning occurs in the system, and most often it's via a simple linear regression model. One of the most important parameters in reservoir dynamics is the spectral radius, or alpha radius. And this parameter corresponds to the first eigenvalue of the adjacency matrix of the network, and it represents a scaling factor of the whole matrix. If alpha is lower than 1, we say that the network is in a stable state. And when the system is in a stable state, the network is good at maintaining memory of past inputs, and similar perturbations leads to similar activations. And the influence of past input fades slowly over time. In the chaotic state, on the other hand, when the alpha radius is larger than 1, tiny variations in input lead to dramatically different states, and the reservoir becomes extremely responsive and sensitive, but unpredictable. And this leads to the memory of past inputs to quickly become distorted. Criticality is the state when Alpha is close to or equal to 1, and it represents the boundary between order and chaos. And at this critical point, the network achieves maximum information processing capacity, and it perfectly balances memory of past inputs with sensitivity to new inputs. And small inputs can cause meaningful controlled changes in the system. Most often, it is assumed that the brain operates at criticality or close to it, and past literature mainly has used alpha as close to 1 to evaluate network's computational capacity. So reservoirs can be used to perform cognitively relevant tasks. For example, two very commonly used tasks are memory and prediction. In the memory task, the signal is shifted backwards in time. And in our case, we are arranging it from 1 to 30 time points. And the network's task is to maintain memory of past inputs. Whereas in the prediction task, on the other hand, we do the opposite. We shift the signal forward in time. And here, the network's task is to match or predict future inputs ranging from 1 to 30 time points. As a measure of the network's performance, we use a computational capacity score, where for each time shift, we assess how much the predicted signal matches the target signal and sum this across all 30 time shifts. So the reservoir is usually a random network that can be used. Many networks can be plugged in it. But what we are doing is plugging the human connectome in the place of a random network. And to do that, we used DTI connectomes from the Center for Attention Learning and Memory. We parcellated the brain with the BrainAtome Atlas with 246 ROIs, and we chose our inputs and output nodes by using the EO7 networks parcellation. And we used the nodes of the visual network as input and the somatomotor network as output. We used the Mackey glass chaotic time series as the input signal and the hyperbolic tangent function as the activation function. And we had the networks perform the memory and prediction tasks. So we know that the brain is organized or optimized for specific task performance under energetic constraints. And we assume that with the correct parameters, the empirical connectomes will have a better performance compared to a network of the same size and density with destroyed topology. And the question is, can we make the connectome-based reservoir networks sensitive to the subtle individual topological differences we observe across human connectomes? And the goal is to have easily manipulable models that can help us to relate, for example, task performance to cognitive variables, or find differences in performance between health and disease. So we came up with two measures: sensitivity to topology, which represents the difference between the performance of the original connectome and a degree preserved randomized model, and sensitivity to weight location, which is the difference between the intact connectome and a topology preserved weight randomized model. Not topology, sorry. Yes, topology preserved. So the topology is preserved, but the weights are shuffled. And before I go on to the results, there are two important papers in the field of neuromorphic reservoir computing. The first is learning function from structure in neuromorphic neural networks from the group of Bratislav Misic. And here, Suarez and colleagues found that empirical connectomes outperformed randomized null models. And on the other hand, the Micheli and colleagues later found that the more randomness allowed in the networks, the better performance the networks achieved. Now, why would these two teams come to such different conclusions? And the answer might lie in what you may notice on this slide, and that is that if you look at the performance, if you look at the last axis and look at the performance, you can see that there are many parameters that lead to different performance scores. And the way to read this plot is that each line traces through different choices of parameters. For example, we can select a certain density or certain input scale, and based on these selections, we might get very different performance of the same networks. And what is even worse, the performance of the network doesn't correlate with sensitivity. So here we can see relatively high performance and positive sensitivity, which means the empirical connectomes outperform the shuffled ones. And we can also observe high performance with negative sensitivity, meaning the randomized models perform well, but they're better than the empirical networks. So going back to this slide, the authors of the two papers essentially chose different parameters, which makes sense since their work had different goals, but it's a good example to illustrate how parameter choice can lead to different conclusions. And one of the most important parameters to choose is the input and output nodes. And in this case of Suarez and colleagues, they chose to use the constrained input and output nodes based on resting state networks, whereas Damicelli and his colleagues chose to use all of the nodes as input and all of the nodes as output. And in our work, we show that this leads to a difference not only in performance, but also to sensitivity to topology and sensitivity to weight location. So if we look at the performance of the networks with the visual network as input and the somatomotor network as output, we can see that it peaks in performance at criticality and shows positive sensitivity to topology and weight location, meaning that the empirical networks perform better than the randomized models. Whereas if we look at the red line, which represents the network that had all nodes as inputs and outputs, we can see that it doesn't display a different performance across dynamics. And it also shows no sensitivity to topology or weight location. Another parameter that we looked at is density, as the first thing one might do if they want to do neuromorphic reservoir computing is to threshold the network. And intuitively, this might seem important, but if we observe the task performance, we can see that if we ignore the red line, which is the lowest possible density to still connect all the nodes, density does not matter that much. But if we look at the sensitivity, there is still positive sensitivity to topology, with most densities being above 0. It's a bit different for sensitivity to weight location, but this varies so little that probably is not that important. It is very similar if we look at prediction. A small difference here is that we can see slightly lower performance earlier at density 0.02. And for this density, the sensitivity to topology is negative compared to memory, where it was close to 0. And sensitivity to weight location is only positive for the highest density. So we can say that network density, even though intuitively might seem like an important parameter, only makes a difference in the extremely low density cases. In contrast, something that sounds innocuous, like the input scale, showed to be quite important. When we look at task performance for input scale, we can see that input scale of 1 clearly outperforms the other two. If we look at the blue and red lines, we can see that for these input scales, the critical point is shifted as they reach their maximum performance at alpha 1.4 instead of 1. And this is because the input scale acts as a scaling factor in a less direct way than the spectral radius. But however, shifted peak is still lower than the one for the input scale of 1. When we look at sensitivity to topology and weight location, we see that at criticality, the networks are positively sensitive only at the input scale of 1. And the situation is very similar for the prediction task, where if we register the best performance for the input scale of 1, and this is the parameter value that also leads to positive sensitivity to topology and weight location. So through this work, we show that before doing anything meaningful, like looking at individual differences in task performance or lesioning empirical networks to be able to come to meaningful conclusions about the impact of lesions on computational capacity, we cannot avoid tuning the parameters. And to do this, we can't only rely on task performance, but we have to in advance assess the network's sensitivity to topology and weight location. We also show that some parameters are more important than others. For example, the choice of input and output nodes and the input scale have a strong impact on the network dynamics, as, for example, if we use all nodes as inputs and all nodes as outputs of the network topology doesn't matter at all. And this makes sense because the signal is not even allowed to propagate, but it's examined in a way that in the same place that it enters the network. In contrast, network density is less important, and it is very important to say that there is no one size fits all. Different network parameters will be works. And these are just a couple of examples. In the future, it is important to understand all parameters and how they interact with each other. I would first like to thank Petra Vertes and everyone in our systems neuroscience lab, Duncan Assel, and his 4D lab that closely collaborates with us on this project, Agostan Michalik. He was a postdoc that worked on the project before me. Big part of this work is based on his work as well. He collaborated with a group of Bratislav Misic, and they put out the Quant2Rest toolbox, which is freely available and was used for this work. And lastly, I would like to thank our funder, the Templeton Foundation, and all of our co-authors, collaborators.